Hi, my name is Michael Raschke. I'm working on my PhD thesis at the Institute for Visualization and Interactive Systems at the University of Stuttgart. In this video, I would like to show you our parallel scan path visualization technique. So, enjoy the presentation. Welcome to my talk about the parallel scan path visualization technique. In the next 10 minutes, I will give you a short motivation why we have developed our visualization technique, and then I will present you the parallel scan path visualization approach. After this, I will show you the 3 visualization techniques which are based on the visualization approach, namely the gaze duration sequence diagram, the gaze duration distribution diagram, and finally the fixation point diagram. I will give you an example of use and finally conclude and will show you future work, which is now current work, really cool stuff. The motivation to develop a new visualization technique for eye tracking was our so-called visual elements user study. Questions which we wanted to study in our user study have been: are there general eye movement patterns? Can different user groups be distinguished based on eye movement patterns? We had 3 tasks and 7 types of visualizations. They are shown here. For example, 1-dimensional, 2-dimensional and 3-dimensional coordinate systems. There the task was to identify a given position in the coordinate system. The second task was to compare triangles with respect to their mathematical similarity. And the third task was to find the largest colored category in a tree map, or the largest category in a tree map, or the largest country in a given map. We used the Tobii eye tracking system and we had 30 participants. These 30 participants finally led then to 2,700 scan paths, which we had to compare, and we wanted to do this with a visualization. This was the motivation why we developed the parallel scan path visualization technique. And this technique works as follows. We define areas of interest in the stimulus, and these areas of interest are mapped to a vertical axis. The whole visualization looks like a little bit like parallel coordinate systems. So we have the areas of interest on the vertical axis, and time is running from the bottom line to the top. The layout here is arbitrary. We have chosen this layout here. The first visualization technique which we have developed on our approach is called the gaze duration sequence diagram. And here, for example, you see a result of a participant which performed the first task to read a given position in a 1-dimensional coordinate system. We defined the areas of interest as follows. You see here areas of interest 1, 2, 3 and 4 on the stimulus at the bottom right here on the slide. Then, as I have shown you on the slide before, the eye movements of the participant started at the area, inside the area of interest number 2, then went to the area of interest number 1, then to 3, to 4, back to 3, then to 2, and so on. Time is running from the bottom to the top here, and the whole performance was around 11 seconds. The gaze duration sequence diagram shows gaze durations. They can be identified by the length of the vertical lines on the areas of interest axis. This diagram shows a scan path. Gaze duration start and end times can be identified by looking into the diagram. What you also see are temporal sequences of attention changes, as I have shown you on this slide. The second technique which we have developed based on our visualization approach are so-called the gaze duration distribution diagram. The gaze duration distribution diagram is based on a work of Guan and Cottrell from 2007. At the bottom right you see the stimulus. Here the task was to state whether the 2 triangles are congruent or not. On the right you see the heat maps, and on the top right you see the gaze duration distribution diagram. The diagram shows summations, the summation of gaze durations in areas of interest with bar charts at the bottom for each participant. We see the scan paths again, but now not only or not the gaze duration start and end times. We see the middle point of the gaze duration by this black dot here. The gaze duration start and end times can be seen by a tooltip moving the mouse over a certain point here. What we also see are temporal sequences of attention changes. Finally, the third technique is called the fixation point diagram. Here on the bottom right, you see again the stimulus. This was now the task to read a given position in a two-dimensional diagram. We have chosen the areas of interest as follows. So we said, okay, there could be a movement of the eyes, for example, from P1, from the point P1 to the coordinate axis, to the vertical and to the horizontal coordinate axis, as shown in the scan path visualization. And we wanted to study this in more detail, so we defined areas of interest 1, 2, and 3 to find strategies for reading P1 and areas of interest 4, 5, and 6 for reading the given position of P2. On the top right, you see the fixation point diagram with the areas of interest. And for example, you can see here, it's described in more detail in our article, but you can see different strategies. For example, there are participants who began to read at first P1 and then continued with P2. The other one began with P2 and then continued with P1. You can see it in the diagram. As I mentioned, it's described in our article. The fixation point diagram shows single fixations. The frequency of fixations, you see the little dots on every vertical area of interest axis. These dots indicate the fixations. You see the scan path, the gaze duration start and end times could also be found out by a tooltip, and you see the temporal sequence of attention changes. This slide can help you to decide which visualization technique you want to use. You want to do an eye tracking analysis, so you start here. If you're interested in an overview, you can use heat maps. If not, but if you're interested in which order items have been looked at, you can use scan paths. If areas of interest are available or possible, you can use some statistics, like for example transition matrices. If you have a larger number of subjects, you can use our technique. Our technique works up to 5 to 15 participants, depending on the different strategies of the participants. If you're interested in fixations, you can use the fixation point diagram. If you're interested in overlaid information, you can use the gaze duration distribution diagram, and if not, you can use the gaze duration sequence diagram. This slide shows now another application example with 3 points in a two-dimensional coordinate system and with 6 participants. And here you see very nice that all the participants which are selected out of the 30 participants from our visual elements user study, that they used a technique beginning with reading P1, then continuing with P2, and finally reading the position of P3. With some practice, you can read this visualization technique very quickly. So now I want to conclude my presentation. In the last 10 minutes, I presented you the parallel scan path visualization technique. We developed 3 visualization techniques based on our approach. They are called the gaze duration sequence diagram, the fixation point diagram, and the gaze duration distribution diagram. One key feature is the display of various properties of scan paths in the same type of diagram. And this picture shows you our current work We described this work in our article as future work. Now it's current work, really cool stuff. So the main challenge is, if you're studying eye tracking, that you have a large number of participants. You want to group them, you want to select some specific participants out of these groups. You want to compare the groups, you want to use different eye tracking, different visualization techniques. So you interact a lot with your graphical user interface. So we thought, why couldn't we use 2 HD screens, large screens, together with a tabletop to interact with this large number of participants? This is current work, really cool stuff. Thank you for your attention.